My name is Laia Saltor Nunez, and now I'm doing my PhD in the University of Leeds. I'm 25 years old and I'm from Barcelona, Spain. When I was 18, I started to study in the Universitat de Barcelona, where I obtained my bachelor degree in chemistry and my master degree in organic chemistry. Moreover, during my third year of uni, I became a member in Oligos research group, under the supervision of Professor Ana Grandas and also Dr. Jordi Agramunt. After my master degree, I moved from Barcelona to Leeds to start my PhD project in the University of Leeds, and by this way, discover the world of sugars. For that reason, I became a researcher in the project ESR2 under the supervision of Professor Bruce Turnbull and Dr. Ralph Richter. Let's take a look into an endothelial cell. Being more specific, the cell membrane and its extracellular space. The glycocalyx present in this interface includes all the structures which contain sugars, so that means glycoproteins, glycolipids, and glycosaminoglycans and proteoglycans. The glycocalyx has different functions, like protecting the cell from external agents or mediating the communication between cells. One of the sugars present in the glycocalyx are Lewis Y antigens, responsible for the different types of blood, A, B, and finally O. On the other hand, there is a bacterium called Vibrio cholera, which secretes the protein cholera toxin. This protein is found in untreated water, infecting humans and being responsible of a mortal diarrhea. If we take a look into cholera toxin, we can see that it's formed by two domains. The A subunit, toxic for the cell, and the B subunit, or CTB, helps cholera toxin enter into the cell. This help is provided by two binding sites. The principal one, which interacts with GM1 ganglioside, and the secondary one, which interacts with Lewis Y sugar. In the cell membrane, there are structures called mucin. These structures are very complex and contain various sugars. Also, they can interact with lectins. The aim of this project is to construct mucin-like structures which have a well-defined structure. To achieve this purpose, the polymer will be derivatized with Lewis Y to study its interaction with CTB in its secondary binding site. But how can a mucin-like structure be built? To answer this question, it's required to understand that there are two parts in these structures, the polymer and Lewis Y sugars. The Lewis Y cannot be linked to the polymer. For this reason, a chemical click reaction is needed to link the polymer with Lewis Y. So it means that a bi-orthogonal reactive group will be added to the polymer as well as to the Lewis Y sugars. Then it will be a reaction just between them, achieving a well-defined single structure, a polymer derivatized with Lewis Y sugar. The chemistry used to make this linkage is inverse electron demand Diels-Alder cycloaddition, EDA. This reaction takes place between a tetraene and electron-rich dienophils, like norbornene, the structure in yellow. It presents a lot of advantages, like high selectivity, fast speed at low concentration, metal-free catalysis, and its compatibility with physiological conditions. An extra advantage, useful for this project, is that in the reaction between a tetraene and a norbornene, there is a change of color, from pink to colorless, so it's easy to follow the reaction by bare eye. Using the other reaction, the polymer will be derivatized with tetraene to make a tetraene polymer. On the other hand, Lewis Y sugar will be derivatized with norbornene, obtaining norbornene Lewis Y sugar. Once both components are synthesized, the other reaction will be used to make a Lewis Y polymer. This plan presents a lot of advantages, like its versatility. to attach other sugars in place of Lewis Y, like Gb3, to achieve a Gb3 polymer